we will start again a little bit late due to the fact that 2 coffee machines for 300 people is not reasonable. Moreover, after a long walk to go there, so we run a little bit late, but we can delay for the lunchtime and then after also for the afternoon, because finally the last speaker, Machado from Ethiopia will not come, and he's not able to present remote, so we will have a cancellation of this last talk. I have also one message for you or your families. If you come with your family, there are transport tickets, free transport tickets, which are available at the desk, at the registration desk. So with these tickets, you can run in Lyon freely for 1 day, but you can take another one the day after, I think. So it is offered by the regional authorities. So if you are interested to visit Lyon or to go in downtown, you can go and take these tickets. So if we can start again for the session, Jean-Louis and Anne. Yeah. Maybe okay. You can. It remains some people in the coffee, but only a few number. So thank you.
Okay, thanks, Philippe. Now we will move to the next speaker. It is Anouk Van Breukelen. She is a PhD candidate working in the domain of animal breeding and genomics at Wageningen University Research. Her project is reducing methane emission through animal breeding, especially in the case of dairy cattle. Today, her presentation will deal with the genetic control of ruminant methane emission. So, Anouk, the floor is yours. You have 15 minutes, and after we will have 5 minutes for questions.
Okay. Well, thank you. Welcome, everybody. My name is Anouk Van Breukelen. I'm a PhD student at Wageningen University, and today I will share here information on the genetic control of ruminant methane emission in livestock. And as we've seen in a previous presentation just before the coffee break, we think that animal breeding can also have a big effect on reducing methane emissions of our cattle populations. And this is still sometimes overlooked. So today I want to give you some more information on what role breeding can have in the future. So, well, I'm coming from the Netherlands, and the research that I'm doing is mostly focused on mitigating methane emissions of dairy cattle in the Netherlands. And, well, similar to many other countries, the Netherlands has very strong mitigation targets with 55% emissions of mitigations by 2030 and to become climate neutral in 2050. And in the Netherlands, it's a bit of a unique situation in that a lot of methane emissions specifically are coming from agriculture. So other industries are relatively smaller, and we have a very big dairy farming sector. And therefore, for on-farm emissions, methane contributes coming from agriculture is quite large. And of these 2/3, 77% is coming from cattle, and in the Netherlands that's mostly cattle of dairy farms in dairy farming. And these are only considering on-farm emissions. They are mostly coming from enteric fermentation of ruminants, so 80% of the emissions are coming from the animal itself, and only a smaller part, around 21%, is coming from the manure. Therefore, in our product, we're also targeting these enteric emissions. So they are produced by the cow when the cow is eating its feed. It comes into the rumen, where enteric fermentation is taking place by all kinds of microbiota. Most of this is during rumination being belched up by the cow again directly from the mouth into the air. And a small part is taken up in the blood of the cows and coming out of the lungs, so also out of the mouth of the cow. And only a very small part comes out of the back end, but by largely the majority is coming out of the mouth of the cow, which has important implications for the measurements we need to take or how we measure methane, which I will come to later. Well, the role that we see of genetics, we know that there is a variation between individual cows. Well, this variation can have different reasons. Some is because different cows might have different diets, they eat different things, and thereby the fermentation processes are different, and the cows emit different levels of methane. but also many studies have shown by now that there's a lot of genetic variation, so genetic differences between individual cows that are accounting for these methane emissions. So estimates from the literature show that between 11% to 43% of all the variation in methane emission between individuals comes from the genetics of the cow, and that's what we call the heritability. Well, therefore, we do believe that animal breeding is can be a very good mitigation strategy. So thereby we select cows that have breeding values for low methane emissions to become the parents of our next generation, and then the next generation of animals will have, on average, a lower methane emissions than our previous herds. Well, a little bit of background on what we do with animal breeding. So that comes back to the quantitative genetics theory, meaning what we see for traits like methane, but it's similar for traits like milk production or height, is that methane shows continuous variation. So as you see in this distribution on this plot, most of the cows will be around the average, with only few individuals that have very high methane emissions and few individuals that have very low methane emissions. And what is actually causing this variation? Well, it's not like a trait, for example, color of your eyes, which is well known. You have maybe one specific gene that will influence if you or cause that you have blue eyes or brown eyes, but for methane emissions, this variation is caused by a very large number of genes with all very small effects. Meaning with this infinite number of genes, it's very hard to pinpoint which genes are actually having an effect. That's because it's such a large number, it's impossible to be able to pinpoint that. And therefore we have to use different techniques, and that's where quantitative genetics theory comes into play. So we look at the whole population, and by measurement of the mean emissions of these animals and the variance or the variation in emissions, we can come to selecting which individuals we expect has a breeding value for lower methane emissions or higher methane emissions. Well, but to be able to estimate these means and these variances, it means that you need to know the methane emissions of a very large number of animals before you can make these selection decisions. So before we can breed for methane emissions, we need to start measuring these emissions, and that is essential. What we are focusing on as measurement systems, currently in our project, we are using mostly sniffers. Sniffers are a device that are placed in the milking robot on dairy farms. There's a tube leading into the feed bin, which measures continuously the concentrations that cows are breeding out. However, sniffers have a very large downside in that they are not very accurate. They are not sucking up the complete air of the cows breeding out. And also the cow can move around in the milking robot, taking his head out of the feed bin, causing inaccuracies in your measurements. But the big advantage of sniffers is that you can measure as much cows as go into the milking robot, and it's more cost effective. Well, another system that we are also using in our research is the green feed, which was shown in the video this morning by the company how it works. So advantages of the green feed are, like you could also see there, is that they use a flux method. All the air that the cow is breathing out is being sucked up by the system. It records where the head of the cow is. However, the downside of the green feed is that it's more costly. So if you want to measure thousands of animals for genetic evaluations, that means that will be difficult to do with green feed units because it comes at a large price. In the literature by other research groups, there are also a large number of different systems that are used. Just shown here quickly, for example, laser methane devices. They are a handheld device, so they are accompanied with a bit more labor. The SF6, this is a tracer gas method where cows wear a backpack with this tracer gas to be able to estimate emissions. Restoration chambers, this is the golden standard, so the cow is locked into a room, all emissions going in and out are measured, so it's very accurate, however, also very expensive. You can only measure one cow up at a time, usually, and the cow has to be locked in there for a longer period of time. So that makes it also difficult to measure large numbers of cow. Other kind of chamber are portable accumulation chambers. So a cow or a sheep or animal is put into this chamber, and then also the emissions going in or coming out of that later are measured. It's a bit less expensive than a respiration chamber and can also be used to take spot samples. Then there's also proxies. So for example, some groups are using milk infrared spectra They are using the correlation or interaction between components in the milk and methane emissions, or measures of microbiota, so taking samples from the rumen and to have some correlations with what type of microbiota are in the rumen and how they are correlated to methane emissions of those individuals. But most important is that if you choose any of these measurements, you need to have a sufficient number of measurements to be able to estimate breeding values for animals. So what we do, or what we are aiming to do, is to have a reference population of animals of which we know their methane emissions, we've measured methane emissions, and we have the genotypes. And then from that we can get prediction equations, and then we are able to predict what kind of breeding value, or how the effect in breeding will be of that animal in a large population, even if it only has information on its DNA, but it doesn't have to be measured itself. And those we call genomic estimated breeding values. Some colleagues of mine, so Yvette de Haas et al., did a simulation study for if we want to get reliable predictions using genomic estimated breeding value, how much data will we actually be needing to be able to make these reliable predictions. So they did a simulation, and before we started measuring on farms, they looked at if we want to get at least 40% of a prediction, which will be maybe required by a breeding organization, how much data do we need? So they came with that we need at least 100, or be recording with 100 devices in 100 hertz, and need at least 2 years of recording. So that is a large number of measurement systems that you need, a large number of farms that you will need to visit, so quite a large project. That is what we are currently doing. So we've started measuring around 2021. So for some farms we are now reaching the 2-year limit of measuring, and we are measuring methane emissions with sniffers on 100 farms across the Netherlands. So here on this map you can also see that all the blue dots, they are different farms, and we are really measuring throughout the country to acquire enough data for genetic evaluations in the end. Then a little bit of what we expect breeding can bring us. So on the figure on the left you see the current trends. So methane emission is currently going up. The total methane production is in grams per day. However, if we look at methane intensity, so that is based on production, it's already going down. So the grams of methane that cows are emitting per every kilogram of milk that is produced. And that is because we are breeding cows that produce more milk, are more efficient, and thereby already the methane production per kilogram of milk is going down. However, this is not enough to reach our mitigation targets. And therefore they also looked at if we have an extra selection step on cows with lower methane emissions, how much reduction can we then actually achieve? In the right figure, the top line, that one is the without extra selection of methane, where we see that the grams of methane per kilogram of milk was going down. But if we look at the dotted lines, so there we have extra selection on lower methane emission, we see that the methane emission in 2015 will even decrease further. So without extra selection on methane mitigation, but only increasing efficiency of production, in 2050 we would have a reduction of around 13% of methane emission, whereas with the theoretical maximum, so maximum selection on methane mitigation, we can reach in 2050 up to a 29% reduction. So that's more than doubling the mitigation of methane emission, which has, of course, a very large impact. What we also see is, well, not only we are working on reducing methane emission with breeding, but there's worldwide interest. So there's many research groups and breeding companies working on this topic. Research that I found very interesting is application with breeding program with sheep in New Zealand. So first they did a 4-year experiment where they had 2 selection lines, one with sheep with high methane emission, and they bred them to be even higher methane emission, and one group with sheep with lower methane emission, they bred them to have low methane emission. And after these 4 years, the difference between those groups was up to 12%, which relates to a difference in 1.3 grams of methane per kilogram of diameter intake that the sheep eats. So it had a very, very big impact. And because of these positive results, they've also been applying it in breeding programs for sheep since 2018, which is very well picked up by both government and industry. And also they're currently working on applying a similar system for cattle. In Canada, they are also, well, since this year have been implementing breeding values, and they are using milk infrared spectra predicted methane, but that is just currently starting, so we still have to wait for the results. And well, similar, we hope to have our first breeding values for methane available in 2025, and we're working hard on implementing that in the industry. But still, there is a lot of research around breeding for methane that still needs to be performed. Very important with breeding for methane is that we apply balanced breeding and breeding goals. So if we mitigate methane emission, we don't want to have a negative effect on other cow traits. So say, for example, we breed for cows with less methane emission, but in effect also the cows start to produce less milk or be less healthy. That is something that we want to avoid at all times. We want to mitigate methane emission while still having good production, good health, good conformation, good fertility. That's why we apply balanced breeding, but to be able to do so, we need to know the relationship between methane emission and these other traits. And that is still research that needs, that is more needed and is still ongoing, because also there large data sets are required which have information both on methane emissions, on milk production, feed intake, and all those factors. And that are also some objectives that I will still target in the next years. Oh, some take-home messages. We believe that animal breeding can contribute to a permanent reduction in livestock emissions, and well, our first results can already be seen in practice, and will be measurable, hopefully, also in the coming years. A lot of research is still needed and ongoing. For example, the correlations with other traits, the accuracies and interchangeability of different measurements methods, as there are so many on the market, and many countries are measuring with different systems. And therefore I'll also focus on sharing data. How can we make the data between different countries comparable will be something that will definitely be needed to target. And these discussions are also very important. Well, if you want to know more about our research, I will present also in an EAP session tomorrow afternoon to share more about the work that we are doing in the Netherlands. And then I want to thank you very much for listening.
So we have time for some question. Matthias?
Thank you very much. I have two questions. The first question, if you gave a prediction about the development of methane per kilogram of milk, right? But if you don't know the correlation between the two traits, how did you do this?
There have been some preliminary studies with a correlation between methane and milk. So for methane and milk, the data sets are a bit bigger, and it depends a bit on the system and the country. Some correlation estimates, there are still some large uncertainties. So it will be more about getting estimates with more high reliability for specific herds. But there are some estimates out there in the literature.
Thank you. And local breeds, what are we doing with local breeds in terms of breeding?
You mean other breeds or?
Smaller breeds.
Yeah, that's a good question. So the research we are targeting now is on Holstein mostly, and the general prediction will therefore also be only available for Holstein. So for other breeds, that would mean that also recording needs to be taken place in other breeds. And there need to be more focus on that.
One question here, Claudia.
I was going to ask a question about trade-offs with other traits, and you haven't gone a lot into this. I remember seeing some research that it actually has an effect on the rumen, and the rumen actually becomes smaller in the low methane producing cows. So as a nutritionist, I would conclude from that that animal probably will need to eat actually more and will actually end up producing more manure in order to have the same production. Can you comment on that?
Yeah, it's not something we have looked at cattle, but I'm indeed familiar with the study. So it's in sheep that they showed that the sheep with lower methane emission, they would have smaller rumens. But how that relates to feed intake, from what I remember, is that those sheep would start to eat more often during the day within smaller quantities. So how it influences exactly the manure, I don't remember, but of course these are factors that we also should keep monitoring. So what effect does it have on the animal, also relating to health of the animal and all of that.
Another question here.
Tannins, University of Wisconsin-Madison. Since the culprits are mostly methanogenic bacteria, is there any work going on genetic editing of bacteria to go directly to a target as opposed to use the indirect route of breeding?
Yes, there is work going on. In our group, it's also just starting, but other groups are further ahead. So, yeah, but they are also looking at incorporating first, not necessarily changing the bacteria, but also breeding might have an effect on the bacteria or microbiota composition in the cow itself. But exactly using directly the microbiota to change the cow, I wouldn't know if that's how that works.
Another question on the left.
Just a quick comment in response to Claudia. So the work we've done in sheep shows that the low methane sheep actually are the same as the controls. A high methane sheep has a big rumen, so it's been slightly misinterpreted. There is a poster here for CT scanning.
I'm Zölkner from Austria. You talked about sniffers versus green feed. How well are those results comparing, or like, can you do one and bring it to the other scale or something?
Yeah, that's also something that we investigated. So we estimated correlations between our measurements that we took with sniffers and the data set that we had with green feed measurements. Phenotypically, it's very hard to compare. The correlations were just moderate, but genetic correlations, they were a lot higher. They were up to 0.76. So for breeding programs, that means that if you would select using sniffers, then you would also see the same result if for the measurements or the cows measured with green feeds.
Thank you about your good presentation. I'm Sayang from INRAE. Hi, I have a question about the hereditary power. I wonder that kinds of feature about related to the methane emission can transfer to the next generation or not.
Can you maybe repeat your question? I didn't hear it.
I'm sorry about my English. It's not good.
So the question is heritability of the selection of the criteria.
Right. Yeah, so the heritability, that does indicate that yeah, we assume that yeah, for the next generation, yeah, the methane will be reduced. I'm not sure if I understood the question.
So I think no more question. Okay, so thanks for your presentation. So we will move to the next speaker.
So the next speaker is Dr. Claudia Arndt. Claudia is leading the Wageningen Research Center of ILRI, the International Livestock Research Institute in Nairobi in Kenya, where researchers are working on establishing verifiable emissions for livestock and improving practices. Before that, she was working as an associate scientist on sustainable livestock systems in Costa Rica and the University of La Molina in Peru. And the floor is you for half an hour. Sorry, for 15 minutes, Claudia, please.
Okay, thank you very much, and thank you everybody for being here. So, good morning everybody. I'm going to talk about effective strategies to mitigate methane emission by ruminants. So I wanted to start out first to see where's the methane coming from. So we have two sources from the cow or from cattle or small ruminants, which would be the enteric methane. We also have manure methane. That depends on the storage system. If it's anaerobic, you will have more methane emissions. But the talk today is going to focus on enteric emissions. And enteric emissions are produced in the rumen of the cattle by microbes. So it functions sort of as a fermentation vessel where we have the microbes that then help the cow to digest the feed that it's taking in, such as the forages or grasses that are indigestible by humans, and then they produce products that we can eat, right? And byproducts of that are methane and carbon dioxide, and the main products for the cow are the volatile fatty acids and microbial protein. So the methane now escapes through belching. often. Do I hold this here? Okay, sorry, the pointer is not working. So it goes through belching, so over 95% comes out from the front of the cow. And then in the rear, in the column, we also have a little bit of enteric fermentation. And then 5% comes out through there. It's reaching my hand, but it's not reaching the screen. So we're focusing mostly on the enteric fermentation that comes out of the mouth of the cow. We're looking at methane emissions, and it's been discussed a little bit already, but like it is about 30% of methane that is produced by humans comes from livestock enteric methane. And methane is a short-lived climate pollutant. So that means that methane has a lifetime of about 12 years. So any reduction in methane we do right now, we can actually feel the difference in 20 years. So if we want to stay within 1.5 degrees Celsius global warming, or 2, we have no other option than reducing methane emissions right now. That doesn't mean that we don't need to reduce other emissions. As Chris said this morning, we also need to reduce our CO2 emissions. It's not just the livestock. We need to do all of them to actually limit global warming. And because methane from cattle is such a big contributor, we do need to reduce enteric methane from cattle as well. So I wanted to bring for a perspective when we're looking at reducing these emissions that we need to look at different scenarios. We need to look at low and middle income countries and high income countries. So I'm giving as an example here Africa and Europe. And first in the table you see actually what the per capita consumption of red meat and milk protein is in 210, 230 and 250. You see that for Africa and Europe. And you actually see that Africa consumes less than a third of what Europe is consuming. So we see a big difference in nutrition in animal protein consumption. We actually have in many parts in Africa under consumption of meat. In the cities you may find an overconsumption, but in rural areas sometimes not so much. And then when we look at the challenge to come, we see the graphs underneath. In 2010 and 2030 and 2050, we will see a huge increase in human population in Africa. And we will also see, because of this, mostly driven by population growth, a higher demand for animal protein. So we see the challenge of African countries is actually they need to ramp up production, and Dominic had already said that as well, because of this population growth. Meanwhile, when we look at countries like Europe, we're not looking at a big population increase. We see like a 4% increase over these years each time, and only a 6% increase in demand for animal production. So we're looking at a very different challenge in Europe while we're maintaining sort of production there. In Africa, we really need to start producing more. And just this is a map, and I have it here just for you to understand where cattle are. So, and where I have now 2000, I'm going to show you 2010 and 2021. When you now look at the low middle income countries, when you look at South America, when you look at Africa or Asia, you see that it gets darker because cattle numbers will be increasing. So we've already mentioned that there we actually will see an increase in population and an increase in emissions. Meanwhile, Europe and North America have been sort of staying the same, but they're already also very high in animal numbers. We now look at the productivity. On the left side is milk per animal produced. On the right side is cattle meat produced per animal. You also see that we have a fairly high per animal production in the US and in Europe, and only for a few low middle income countries you actually have a high productivity. For the others it's quite low, and we're looking at quite low productions of animals still. We're also looking at extreme differences of production systems. So I wanted to show you really the two extremes. So on the left we see milking parlor, rotary milking parlor in China of a dairy that has merely 500,000 cattle. And on the right you see hand milking, which is done in Uganda, of somebody who has 4 cows that are milking. That doesn't mean that you're not going to find that Uganda picture. You will find that in China as well. It's actually, I think in China, maybe you have these extremes in productions in a very close relation. And when we think about mitigating methane emissions, we need to understand the systems we're in and the differences in these systems. And to talk about the mitigation strategies, I'm talking about our paper that was published last year in PNAS. Some of my co-authors are here as well. I thank them very much. It was a long process until we got there, but we finally got there. We did a meta-analysis. We looked at nearly 100 mitigation strategies, and we wanted to see which ones are really effective and where we do have enough data for. So we were really rigorous in picking the ones that we finally recommended, because we wanted to make sure that these mitigation strategies are really working. And the other purpose of this paper was to show what can we actually do with these mitigation strategies now? How can we actually meet the 1.5 target by 2030 and 2050? So we found in total 8 mitigation strategies. We did classify the mitigation strategies into mitigation strategies that would reduce emission intensity, meaning emissions per product, and also increase animal productivity, and mitigation strategies that would reduce absolute emissions, as well as mitigation emissions per product, but have no impact on productivity. And we were not accepting mitigation strategies where we actually would see a decrease in production, because that is not really a definition for us for a mitigation strategy. So when you see here, we had 98 mitigation strategies. The blue mitigation strategies on the top, these are the ones that reduced emission intensities and increased production. So on average, these 3 strategies would reduce emission intensities by 12%. And these strategies are often more relevant for low productive systems. It's like increasing feeding level. This means that in systems where we have low forage availability at times due to season, we see that animals are fed suboptimally, so we need to overcome that. So this is really a message to that. We also need to have decreasing grass maturity, so we need to get to an optimal stage with the grass maturity to have optimized production, and also feeding some concentrate can help. We can feed it very high, it influences the pH in the rumen, will impact the methanogens, but even in systems where we have low production, there's often energy, and more often even nitrogen is lacking. So if we can substitute the missing nutrients, we can make big gains in productivity gains. For the absolute mitigation strategies, we found the most promising were the methane inhibitors. We had Bovear there, which was showing the 30% reduction throughout consistently. Tanniniferous forages, also had electron sinks and oils and fats and oil seeds. With these strategies, the problem is that as they didn't really show an increase in production, it is unclear what reasons farmers would have to adopt these, and how we would actually incentivize adoption from farmers to do that. It could be by increasing maybe product value, so they can offset the increased costs they would have through implementing these strategies. Now, when we looked at how can these strategies reduce emissions, we wanted to compare that to the IPCC target reductions. So in 2030, we need a reduction by -11 to -30% of agricultural emissions, and by 2050, -24 to 47%. You see the red line here, that's the best business as usual. So if we continue doing what we're doing, and we're not doing anything, and then we were assuming only if we had 100% adoption, which is very unlikely to happen, of 2 of the strategies, the most promising for one system that reduced absolute emissions, and the most promising that reduced the product-based emissions, we would reach the target by 2030. But it would really need 100% adoption simultaneously on all farms, and you know how improbable that likely is. And even if we did that by 2050, we would not reach it. Now, the situation was very different when we look at high-income countries. And this is, again, Europe as an example. we only see the baseline here is increasing by 11%. So with these strategies implemented, we actually would be able to reduce emissions in the 2030 and 2050 scenario if we really had 100% adoption. And that's because we're fighting a different fight here. We have the cattle already there. We already have the productivity that we need. We're seeing very little population growth. When we look at low and middle income countries, and this is again on the example of Africa, you see that there's a huge increase to be expected in emissions. And that even if we implemented these mitigation strategies, we would curve the increase in emissions. We would not be able to go anywhere close to the targets because we're going from such a different baseline scenario. So it's very important to understand the fights we're fighting and how to go about it. And again, this is mostly through population growth. You saw that the increase in demand by 2050 was still a third of what we would have seen, like the consumption we will see in Europe. So with that, I want to come to my takeaways that we have effective methods to reduce enteric methane emissions, but it's very unlikely that we have 100% adoption, and we actually need to understand what barriers are there for farmers to adopt these mitigation strategies. And on top of this, we do need additional mitigation strategies, and we need to better understand how we can use multiple mitigation strategies at the same time to reach our target, because implementation of only one mitigation strategy is not going to cut it. I also wanted to use the space to advertise the conference that we're going to have in Nairobi in 2025, that I'm going to be chairing, the International Greenhouse Gas, Animal and Agriculture Conference. So I hope you put it into your calendar and I see you there. And with that, I want to thank you very much and happy to entertain questions.
Thank you, Claudia. Any question? Jean-Louis, please.
Yes, I have one question. Among the different levels of action, you mentioned the ratio of forage to concentrate, but we can have a trade-off between feed and food after. So this level is very efficient, provided we have a huge response of milk yield per kg of concentrate. What is the response of milk yield per kg of concentrate in low and middle income countries in Europe? Because if we produce less than 1 or 1.5 kg of milk in addition per kg of concentrate, we will increase the feed-food competition in the same time. So
I can't give you the figures for that because I don't know them, but like it would be much higher because we're looking at a cow that produces on average 4 kilos of milk a day. So if you put a kilogram of concentrate in there, that is the right composition, you can get quite a bit of increase, right? And then the emissions per product, they're going to be more affected when you have a low producing cow, right? Because right now, if you double, like if I have 4 kilos and I put another kilo, then I'm increasing milk production already by like 25%. Yes. So the reduction in emission intensity is quite high. If I increase a kilo in a cow that produces 40 kilograms in percentage of emission reductions, it's not going to be as big. And also, it would help more with food security, because I didn't say that, but I actually wanted to. The thing is, because sometimes we're hearing like, why don't we produce it somewhere where we are so much more efficient and where we have so much less emissions per product, and bring it into places to Africa. And honestly, the people who need it in the rural areas, they're never going to be able to pay for these. Like the people in the cities will be able to get it, but for actually having food security and saving food security, importing is not the solution. And what we saw during COVID as well, that you have production chain like disruptions, how we're going to, like, COVID was probably not the last time we see that. I hope really don't have to see that again in my lifetime, but I'm not so sure about it. And that kind of scenario may happen again. So I also think it's very important for countries to be self-sufficient.
Dr. Arndt, thanks very much for the very comprehensive and very encouraging approaches. You mentioned the factor of acceptance that would not be possible to be like 100%, right? But the acceptance really very well correlated with the economics and the inputs of the possible interventions technology. So whether you have evaluated, these aspects, whether, among the possible technologies available at the moment. And I think that there's a need to work, more immediately with these strategies in order to reach, sooner, and also to be sustainable, like I mentioned this morning. That is very essential and very important.
Yeah, thank you. No, I agree. I agree with that comment.
Please go ahead.
Yes, thanks for your great talk, Rafael Muñoz Tamayo from INRAE. So, as you mentioned, there are some studies that show also the relationship with methane and feed efficiency. And I wonder if I would like to know your opinion about the potential relationship existing between the animal robustness and methane, because I think there are few studies exploring this area, and I would like to have your opinion. Animal resilience, the capability of an animal to face perturbations, for example, if you have any information about this aspect.
Can you reformulate the question?
Yes. I would like to know your opinion about potential links between methane emission and animal robustness,
and
animal resilience.
Seaweed, yeah?
Resilience.
Resilience. Animal resilience.
Yes. Sorry.
Okay. Okay. So that was for low middle income countries that are disproportionately hit hard by climate change. That's very important. In these regards, we're looking a little bit more also on the animal health interventions that have been mentioned before. In this study, we didn't have enough data on animal health studies because there wasn't really a lot of interest in funding these, and they're quite complex in the past. But there are certain interventions like better animal nutrition, better animal health, that will also increase the resilience of these animals, and also genetics in that regards, right? If we have an animal genetic that's more adapted to the environment and to the feed that's available, they will make it through the rough times. I know for Kenya we had the huge drought, and I had the State Department of Livestock commenting like the native breeds of sheep made it, but like a lot of them who had exotic in there because it was such a long drought, they didn't make it. We lost so many animals. So I think we already have a lot of the genetics, but we also need some on the production side as well. And also not just on the animal, also on the plant to improve that and improve the nutrition, because we can't improve just what I've seen a lot of animal production improvements through breeding, but the nutrition and the animal management side improvement didn't go hand in hand. And then you're standing there with a cow that could produce so much, but there's no nutrition to make this, enable these animals to do that.
We're going to take two last quick questions, please. First here, and then Philippe quickly. Thank you.
Hi. So I was wondering, I was having a question regarding one of the strategies that you have mentioned that is used to reduce methane emissions, which is feed. And I was wondering if when people are putting in the model these feed inhibitors, are we considering the emissions that are produced while producing the inhibitors?
We're looking at that. So it depends on what additives you're feeding, right? If you have boviar, I know there's not that much emissions associated with this production. But if we're looking at seaweed and aspergillus, which we didn't consider in our study because at that time there wasn't enough research published yet, we're looking at a very different scenario. So overall, this will need to be evaluated then in life cycle analysis, because you're also going to have other trade-offs with like nitrogen emissions and so on, and we shouldn't just limit ourselves to methane there.
Yes, no, I mean, I agree there's not enough data. I was asking because I'm working on the feasibility study of purifying some plant components to produce inhibitors. And of course, at the moment I'm concentrated on see how much they reduce methane emissions, and it's very promising, but inside me, I'm thinking, because I can see the big bioreactors that need to be used in order to do that. So I'm thinking, till which extent?
Yeah, but I think there was where we really need to go with life cycle analysis, and you need to look at the trade-offs, because at some point we don't want to produce more CO2 or nitrous oxide either. Exactly. But in the short term, we also need to get methane really down. So that really will need to have a detailed look, and also the costs associated with it. But also I think working on these, isolating these compounds, I have faith that technology is going to get much better, and we're going to get much more efficient in this in future. So if we know what is working, that we're going to get processes that are going to be not as costly and environment with a high environmental impact, and then make it work in the system. Like that's what I'm really hoping for. Because everything is inefficient when we start out, but like if you look what we've gotten through the last 50 years and what technology has done, I think it's good what you're doing, and if we find something, I hope somebody...
No, me too, I have hope. I have hope. Thank you.
Thank you. Philippe, for the last question.
Yes, Philippe Spino, WAAP. Thank you, Claudia, for your talk. After the talk of Anouk Van Breukelen, I was surprised that you don't mention too much genetics and in your different propositions, selection against methane production. Is there any data on among breed variability in methane production breed? When you compare breed, for example, in Africa, which has a lot of breeds.
We didn't include breed in here because there weren't enough studies. Didn't make the cut. We had 2 studies or whatever. The breed, and actually the studies we had, we didn't. There was no effect, but there was really not that many studies at the point. So it's over time, and then to have to look at it again. I know the database has been updated by now, at least on the dairy cow side, and there was a data and briefs published recently. So also if people want to take a closer look at these things, there's a possibility. But back then, I had 3 studies, I think, maybe 4, and then it was different breeds, and it's which one do you put now for the low emission breed, and so on, because it's all different ones. It wasn't a study on exactly the same breeds, so it's hard to combine that in a meta-analysis.
Okay, thank you.
Thank you. So we have 2 more exciting presentations before we can break for lunch. Jean, would you like to introduce the next speaker?
Okay, no, it's my pleasure. Now it's my pleasure to introduce Bart Lardner. Bart is professor at the University of Saskatchewan in the Department of Animal and Poultry Science. He is a specialist of beef production with special focus on forages, pasture management, animal nutrition, and herd management. His research worked closely with the Western Canadian beef and forage producer, and today his presentation deals with the potential of legume tannins to mitigate methane production. So Herbert, the floor is yours. You have 15 minutes, and then we will have 5 minutes for questions.
All right, thank you very much for that, and thank you to the organizing committee for this invitation. I'm going to come at you from a very applied approach and a Canadian perspective in my talk here. So we'll chat about tannins, legumes, and methane production and some of the challenges of integrating legumes in our pastoral systems. So to start off with, alfalfa or lucerne around the world is really the most preferred forage legume in our systems in Canada to improve the nutritional quality of the pastoral settings. And certainly the leaf-to-stem ratio is greater for our legumes versus our grass species, and so therefore we like to have that looking at a significant level of composition in the pasture. And really it's the association between these legumes and that ability to fix atmospheric nitrogen through that biological nitrogen fixation process. And so really, if we can get alfalfa into our pasture systems, there's going to be a huge benefit. And I cite Dr. Bernie Gopelin, the late Dr. Gopelin, in that there was more economic loss of the fear of bloat from grazing alfalfa than actually producers using it and integrating it into their pasture systems, which is a bit of a challenge going forward. And in fact, we were thinking about using alfalfa in our systems many years ago. So McCaughey cited that, yes, if we put these legumes into our pastures, there is potentially the ability to decrease methane production. That's a good thing. So you look at legumes, you look at integrating them into your systems. Obviously, the atmosphere is 78% N. Why not through the symbiotic relationship with BNF and the legumes and the symbiotic relationship? why not have them into your pasture systems? And so my whole program has been looking at integrating different types of legumes, both annual and perennial, in our systems, and certainly to utilize that ruminant for that nutrient cycling and trying to reduce the inputs, the costly inputs that producers are incurring in Canada. We chat about legumes, and we need to also have a conversation about condensed tannins that are found in some legume varieties. And so they're really a naturally occurring plant secondary compound, and they are found to have antioxidant, antimicrobial, and antiparasitic activities, and the ability to precipitate out proteins or to slow down that rumen degradation process. That is what's happening in that alfalfa type situation. With those CT legumes, we don't see that. We see that slow degradation in the rumen to reduce that microbial breakdown, reducing the risk of bloat, which is kind of a safeguard, also improving or increasing bypass protein, and potentially to reduce the enteric emissions from grazing these condensed tannin legumes. Some of them, for example, that we're looking at in Canada are sandfoin, bird's foot trefoil, and purple prairie clover. I'll focus more on sandfoin here in my discussion. Bird's foot trefoil is one that needs a higher rainfall type environment. There's a lot of conversation about purple prairie clover. Why? Because it has a high concentration of condensed tannins. However, in my mind, what's the biomass yield? Is there enough of significance in our pasture systems to provide that benefit going forward? So the two that I'm going to talk about specifically are sandfoin. It does contain condensed tannins, and we've done a lot of research on it, specifically in Western Canada. And the other one I'm going to chat about is sisal milk vetch, and that's interesting legume as well, but it does not have condensed tannins. It has a venation pattern in the leaves, which again slows down or reduces that degradability in the rumen. So what are some of the mechanisms that we see or suggested in the literature? Because of the increasing interest in these legumes, there's various theories put forward. It may be due to providing protection from the rumen microbial degradation process, these condensed tannins, while still allowing some intestinal digestion. The tannins, do they act directly to reduce methanogens, or the process of methanogenesis, or act as a hydrogen sink, because there's a lot of hydrogen ions produced in that rumen fermentation process, or do they decrease the nutrients to the microbes that allow these reducing mechanisms from CO2 to CH4? So briefly, we've heard about what happens in the rumen. Certainly, low quality, high fibrous feedstuffs hit the rumen. The co-products or byproducts are the short chain fatty acids, specifically acetate, propionate, and butyrate. And of course, the gases that are released. We've heard about those from numerous speakers. And it's very dependent on the diet or the pH level in the rumen, the microbial population. These are all certainly involved in the ability to produce those methane products. I also need to echo what I've heard earlier this morning, in that the ruminants are really upcyclers, okay? They take those low quality, high fibrous feedstuffs and they produce high quality protein. And so in my mind, you as a human, you have a choice. You're getting protein from meat or you're getting it from plant, but certainly in my mind, beef cattle are beneficial because we're utilizing marginal lands throughout the world and producing some type of a saleable product. So again, we've seen this process of how methane is produced in the rumen through microbial digestion, producing those three short chain fatty acids, and of course methanogens being part of that process. If you could advance the slide here. Here we go. And what's interesting here, you see that the production of acetate is actually contributing 4 hydrogen ions to this process. The production of butyrate is contributing 2 hydrogen ions to the process, whereas when we produce propionate, we're actually utilizing or taking away 2 of those hydrogen ions from that hydrogen pool. So probably beneficial when we look at the 3 volatile fatty acids. We're really focusing on propionate because that is certainly one that's going to utilize some of those hydrogen ions in the synthesis process. I need to shout out here about our footprint in Canada, the beef industry specifically, looking at a 30-year timeline, and that now, today, from 1981 to 2011, we're producing 15 less GHGs compared to beef production back in 1981. And these are really via technologies that have allowed us to improve production efficiencies in the beef industry. And so you can see a lot more information on this from our website there. So, you know, the production of methane is a huge energy loss, about 12% of gross energy intake. That is significant. And so what are some of the strategies? We've heard some of those from previous speakers. Dietary ingredient manipulation is one, feeding ionophores, some of the algal products that are out there, inhibitors like 3-NOP, but I'm going to focus on improving herbage quality with legumes or condensed tannin legumes. So in my mind, this is probably one of the most effective strategies for our producers in Western Canada, you know, is to assess the pasture composition and the quality, and is there some way we can actually increase composition of our legume species, maybe increase composition of our tannin legumes in that forage stand, and really look at maybe we can utilize those stands in a vegetative type maturity stage versus reproductive maturity stage. I throw this out there because I find myself after a 30-year career being an advocate for the industry. And the two sources of CO2 coming from ruminants, we know there's a recycling process that happens every 11 years through phytochemical oxidation, converting CH4 to CO2. It's taken up by the plants, really a regenerative process, whereas those CO2 levels coming from fossil fuel burning are not going that same pathway. And so we need to be aware of that. Probably in this room, we're talking to the choir, whereas I think we need to reach the younger generation. I think we need to get into the classroom and have these conversations and really start to allow that learning process to go forward. Some of the early work that was done in Canada looking at integrating legumes into pastoral systems, McCaughey back in '99 compared a grass alfalfa system versus a straight grass system, found that binary mixture had greater dry matter intake, and certainly from that they saw lower methane from cows in those binary mixtures. Waghorn, a little later, compared trefoil, bird's foot trefoil in this case, to a ryegrass clover, and observed a twofold range in methane emissions from those bird's foot type pastures. And certainly species selection would play a huge role in reducing these emissions going forward, specifically condensed tannin legumes. Ominski looked at stage of maturity and that grazing early in the season emissions were less compared to grazing later for a more mature forage, 11% of gross energy intake compared to grazing in immature or vegetative, only 6% of gross energy intake. So just to finish up, next couple of slides, we'll talk a little bit about some of our program at University of Saskatchewan. And really, we've looked at trying to integrate legumes, condensed tannin legumes into our pasture systems and see, is there some advantages, not only improving forage quality, but also reducing emissions. The two methods that we've been measuring for the last 6 years now is, we heard about this earlier with the SF6 or sulfohexafluoride technique. It is a laborious system. I have a great team that gets out there and allows our animals to be adapted to these yolks, and over a week time period we'll gather what we feel is pretty significant data. That's the older technique, and of course we've heard a lot about this, the newer technique using the green feeders, hence one of the sponsors of this particular session. This is a study, a 5-year study, where we're comparing annual systems to perennial systems, and I need to make note of my two students, Megan Wasson and Jordan Salmons, and we're looking at 2 annuals, and so that's typically a fall rye clover and then a very simple polycrop, compared to that meadow-brome sandfoin, there's your condensed tannin legume, and a traditional hybrid brome alfalfa type pasture going forward. This is really one of the most comprehensive studies that I've set up in my career. I've got soil scientists, ag economists, engineers, soil microbiome folks on this particular study, so I'm just going to talk a little bit about some of the biomass and some of the animal performance indicators. Looking at these four systems, how did the critters do? Well, you can see that certainly we saw better gains on that perennial hybrid brome alfalfa system. This is after 2 years of data. And then looking at the effect on the systems on forage quality and enteric emissions, proteins were a little lower in those perennials compared to the annuals. NDF values were, I think, quite reasonable going forward. And so we did see lower emissions on those metabrome sandfoin pastures. Finally, how can we get legumes into our pasture systems? And we have a lot of 30, 40-year-old pastures in Canada. And so we've looked at different methods like sod seeding. And this is just a 5-year study that we're working with Tess Mills and Bree Kellen on, looking at different varieties, sod seeding. It really, for me, is persistence of that legume. Will it stay in stand over 4 or 5 years, or will we lose it after 2 or 3 years? And so we focused on sisal milk vetch and sandfoin in this particular study. So just a snippet of some of Kellen's data. In that, yes, we did see a difference in short-chain fatty acid, or A to P ratios, in steers grazing the sisal milk vetch, probably more propionate being produced compared to the sandfoin or the control, which is a straight grass pasture. But we didn't see any effect in terms of methane production in gram per kilo of dry matter intake. So just in summary, legumes, we feel, can improve the forage biomass and quality of our Canadian pasture systems. Condensed tannins do have the ability to reduce bloat and increase nitrogen utilization, and that reduction may occur when ruminants do consume those condensed tannin legumes going forward, but we need to continue to do more research. We need to really look at what is the variety of forage species more legume than grass going forward. I didn't talk about carbon sequestration in this particular talk, but certainly one is going to provide adequate biomass and yield, suitable animal performance, capture as much carbon, and reduce emissions. So with that, I want to say thank you and then make note to my incredible team. Thank you.
So we have time for question. Please, so first one here.
Thank you, Professor. Thank you for your presentation. I'm Ran-Yi, and I have 2 quick questions. For the first time, we do know the tannins is really can have it has the positive effect on inhibiting the methane production. But because the flavor, I mean the trail, the ruminant can feel it. So how to deal with this issue and how to balance that? Does it make sense?
Sorry, I didn't. Can you repeat the question?
Yes, the tannin is flavor. The ruminant can feel that. So they dislike to eat the feed with the tannin. So how to balance this issue? No?
Oh, do they like to graze?
The question is around whether some of the tannins affect voluntary intake because they can have anti-feedant properties. Animals don't necessarily like some tannin-containing plants.
Yeah, okay, so the question is about the level of tannin in the plant material, and certainly that it can be an issue because they will affect palatability depending on the plant species. We have not seen that issue for sandfoin specifically. In fact, the cattle will go and graze that legume out of the pasture, so there's selectivity going on. The challenge with sandfoin is persistence, and so we're seeing it's not in stand after 2 or 3 years, and that's a cost negative for the producer. So hence we're focusing more on sisal milk vetch in combination with alfalfa going forward.
Okay, thank you so much. And the second question is, when the grazing, the clover seems like that, when they inhabiting the methane production, do you think about which is the major reason for that? Is that because of the high fiber feed, or it's have the phytochemical inside? Does make sense?
Sorry, I didn't get that question either.
The clover, maybe it including the high fiber insect, and or it have the most important phytochemical effective.
So the clover had a higher intake or didn't have a higher intake?
Yep. Actually, the higher fiber in the grazing, the feed, that will decrease the methane production. Is that correct?
Well, it was the trefoil, birds with trefoil, and then we had the clover pastures and the trefoil. They saw lower emissions.
Okay, thank you so much.
Just very quickly, you didn't mention nitrogen excretion at all, and particularly using legumes that can have a much higher nitrogen content. Have you also looked at any effects on nitrous oxide emissions coming from perhaps having a greater N intake, greater N excretion, and looking at how that could offset any methane savings from the introduction of legumes?
Yeah, that's a great question, is looking at the whole cycle itself. And on that sod-seeded study, we are looking at that. So our soil scientists are measuring the nitrous oxide coming from the manure. And we're finding, in fact, that, in some cases it exceeds the benefits, in some cases it neutralizes. And so it depends on the percent composition of the legume in stand, which can vary, as I said, depending on how long those legumes stay in stand.
Tricia Johnson, New Zealand. We've heard in a couple of the previous presentations about the need for adoption around the methane space. Have you, with this work you've been doing, looking at the fatty acid profile of the either subcutaneous or intramuscular fat that can vary as a result of condensed tannins, and that could be a win that we could be looking for?
Sorry, I didn't get all of that question, sorry.
Sorry. Have you been looking at the fatty acid composition of, like those of beef cattle you were looking at, the fatty acid composition of intramuscular fat or subcutaneous fat?
Oh, the composition, like doing some ultrasound on our animals?
No, doing fatty acid, like in the dead animal to actually measure.
Yeah, no, that's one area we're not looking at, sorry.
Oh, cool.
One last question.
I'd just like to share the in regards to the concentration of condensed tannin. If condensed tannin is more than 6% of dry matter, that would affect on the palatability and digestibility of the feed. But the overall intake in terms of dry matter intake in animal should not be more than 1 to 2% of dry matter intake. Thank you.
Yes, I agree. Thank you.
Thank you, Herbert. I think we can move on to the last speaker.
Yep.
And please reach out to Herbert if you need to continue the conversation. I'm sure the conversation might be easier without the speakers and the microphone in between. So our last speaker is Diego Morgavi. He's joining us on the floor. He's a research director at the Department of Animal Physiology and Livestock System at INRAE. And his research is in the domain of gastrointestinal microbiology and animal nutrition. And he's in particular a specialist of microbial ecosystem and methanogenesis in ruminant. So also coordinator of this project, Holo Ruminant, funded by the European Union. And the floor is you, Diego, for 15 minutes.
Thank you, Anne, for the introduction, and thank you for the invitation. So I will talk about, again, how to reduce methane emissions through feed additives and feed nutritional type of approaches. And as a difference probably with Claudia, that has set up make a wonderful job in saying the approaches that are useful. I will focus on this on early life. So I apologize also because some of the concepts will be repeated, but probably in different pictures, and I hope that make the point on how important they are. Methane as a global level, the problem is that there are sinks in the natural, and there are emissions that are natural, and the problem is the man-made emissions and the anthropogenic emissions. And as we saw before, enteric fermentation make up to 27% of methane emissions. That of the total one, and if we consider just the agricultural point, is up to 39%. So for the huge number of livestock that we have in the world, they affect planetary boundaries, the land use, the use of freshwater cycle, and also because they emit a lot of greenhouse gases and they affect climate change. But we also saw in other presentations that there are other aspects that we have to consider in livestock production and ruminant production. That's the point of food security, which is really important. And the fact that they provide up to a third of the daily intake in protein. And as you can see in the figure, there's huge differences in high-income countries and low-income countries. So high-income countries, usually if you look to the Lancet diet recommendation, we are eating too much animal proteins. But on the other side, there's a deficit in the protein that is that people eat in low-income countries. From 2020 up to 2030, the increased production is going to be 1.4, is the predicted consumption. And that is not enough to meet the sustainable development goals of zero hunger. Also, if we want to keep methane emissions at the level, or greenhouse emissions at the same level, the only way is to increase production, which we also know that it's not possible to increase 35 or 30% production in just 10 years. So the only way that the world is going is to increase the number of animals. And that brings us the point of methane, as Claudia said, is a short-lived gas. And if we can reduce methane, we can have an impact on climate in the medium term, shall I say. So to reduce global emissions of endemic methane, we need effective mitigation options. And that's a no-brainer. Everybody understands that. But they have to be applicable to different production systems as well. And importantly, also, they have to be adopted by end users. So there's a problem of end users. They have to be willing to use the products. They have to find an interest. They have to be able to afford them if the products are available. So that's one of the big problems that other speakers also pointed out. Just coming to the numbers of animals, it's similar to and the production systems that we have to target. We have to take into consideration that most of the mitigation options are applicable in systems that are quite intensive, and they are applicable in climates which temperate type of climates. So most ruminants actually in the world are in grazing and mixed crop livestock systems, and they are concentrated in tropical and subtropical areas. So you can see here the distribution in livestock units. So one livestock unit for cattle and large ruminants, and 0.1 for small ruminants. And you can see that most of them, as I said, are in tropical areas. It's true, it doesn't work. It's work a little bit. In tropical areas, but this is the data from 61. So in 40 years, the evolution of livestock population has changed, and that's why it's important that we have solutions also for grazing and mixed crop systems in tropical areas and subtropical areas. Which are the strategies to mitigate greenhouse gas emissions? So low emission systems that we are going to probably to see in the afternoon. Everything that improves nutrition, reproduction, that reduce the number of animals that you need to produce a kilogram of milk or a kilo of meat will reduce at the end the amount of greenhouses that are emitted. Animal genetics, we saw that. And the other is rumen fermentation, trying to modulate the fermentation activities or the microbiota. And these are the dietary strategies mainly. Have to remind us, as the last speaker said, methane is a microbial process. I'm not going to go through this, but it's just to make the point that when feeds are digested and goes down to methane, there's a series of processes by the microbiota. And methanogens, which are the only producers of methane, are at the end of this trophic chain. So for feeds in general and feed additives, we can distinguish two modes of actions in which they reduce methane. We have the generalist and a specific mode of actions. And if we go back to the previous picture, so the generalist will have an effect on the whole ecosystem, on the whole microbiota, whether the strategies that are more specific will go either affecting methanogens or affecting the production of methane by methanogens. And the approaches are several, as Claudia said, but there is probably one problem on that point, in which either feeds or additives have to be provided daily to be effective. And that's because the microbiota is very resilient, the methane production is very resilient. There is a functional redundancy also in the microbiota. We know also that is in the adult animals. In young animals, this microbiota is not yet established. And we know that the colonization of the rumen, of the gastrointestinal tract, is a progressive process that goes, the microbiota is acquired at different stages, at different, as the animal develops and as the animals change from liquid feed to solid feed. And we also know from ruminants and also from other mammals that early life events that affect the gastrointestinal tract affect the microbiota composition. And some of these initial colonizers in the rumen or in the gastrointestinal tract can persist when the animal is adult. So the approach of trying to modulate the colonization has been applied for improved health, for improving the production of animals, and also a little bit to decrease methane emissions. And the question is that it's possible. For the literature, some studies in which they use the type of generalists that I mentioned before, feed additives like oils, garlic oil, for instance. What they saw is that after the treatment ceased, so they measured the effect after, when the animals grew up, some months after they were born, there were persistent effects on the bacterial community. Most of them, or all of them, I would say, there was no effect on methane emissions. There's a study with a specific feed additive, bromochloromethane, in which what they saw is a change that persisted in the archaeal community, and there was a change in one of the treatments, in the animals that were treated early in life, the dam and the kid, and after the treatment was stopped, those animals were emitted less methane than controls. And there's another study in which 3-NOP was used, nitroxypropanol, and I will focus a little bit more on this because we have more information. In this study, we use 18 calves divided into groups, and one was treated with 3-NOP from the beginning, from the day they were born until they were 14 weeks old. And we measure methane and other measures on the growth. So in terms of growth, there was no differences in those animals between control and 3-NOP. We can see it here. And there was a slight tendency for the 3-NOP to have a smaller weights, the animals. In terms of fermentation parameters, also there was no difference. I just remind you that the treatment stopped at week 14, so the other weeks the animals were without treatment and they were together in the same group. As regarding to methane emissions, that shows the time from weaning. So the gray line, I don't know whether it can be seen from far away. There's a gray zone here from 1 to 3 weeks. That means from week 12 to 14 of the life of the animals in which they were treated. And there was an effect of the treatment that reduced about 10% emissions. And after that, the treatment was stopped, and that trend was also observed after the treatment was stopped. And when we brought back those animals that were almost 1 year, the differences still were there. It was about, in average, was about 10% or 12%. Because the animals were a little bit lower in weight, we measure also using another metrics, grams of methane per kilogram of body weight, and the differences were still there. So in terms of the microbial communities, there were differences between the two groups. You can see the old samples here, but also separated by weeks. That's in terms of bacteria. We observed the same thing for archaea, in which here in the figure, you can see how the archaea, the evolution according to the age of the animals. And there were some differences here that we can see here in the picture. So those two groups were clearly separated. The same was for rumen fungi. That's the representation for at the end of the study. No changes in rumen protozoa, and there were some differences in bacteria in the fecal microbiota, but not for other communities. So the take-home message will be that it's possible to modulate the gastrointestinal microbiota through early life approaches. And those changes might or can be seen after the treatment stopped. And also, in some few cases, there was a persistency of methane reduction, the phenotype of methane reduction with some feed additives, particularly in those which are specific for methane inhibition. So we changed the pattern of acquisition of the microbiota into the normal one, into a new one using a feed additive. And that, I think, that opens new perspectives for reducing enteric methane emissions, because it offers the possibility that to treat the animals for a short period of time, and if that we can reproduce that using other feed additives or other strategies that could be probiotics or microbiota engraftment, or using the contents of animals which are low emitters, we have probably the possibility to have, during a short window of time, to affect the microbiota, and that, if persists in the long term, that could bring another small solution to the big problem of methane emissions. And with that, I would like to thank the co-workers of this work and the funders. Thank you very much.
Thank you, Diego. Do you have any question for Diego? Yes, please.
Thank you, Diego. Philippe Cheminot from WAAP. I have two small questions. Do you mention the doses of TRINUP that you use, and do you test an increasing doses of TRINUP?
No, we don't remember it by heart, but it's not the same dose that is recognized by the DSM for adult animals. So the dose was given in a liquid form by oral garbage to the animals.
But you test only one dose?
Only one.
And if...
sorry, it was calculated every week according to the weight of the animal.
And my second question was, after 1 year or after 50 weeks, if you repeat the treatment with TRINUP, do you have also a decrease? Continue to decrease, or?
We couldn't do that. Yeah, that is come back to your question before, if you can add another strategy to reducing methane on those animals, that would be nice. But I don't like to focus on 3-NOP here. It's like the concept is whether I think there is something there. We don't know yet how to well manage it, because I know that some other groups couldn't reproduce the same results. But on the other hand, there are other reports in which you can see that early life interventions can change the phenotype of the animal in the long term. So not just for methane. So whether it's just the microbiota that is changing, whether it's the early microbiota installation in the rumen affect, or the gastrointestinal tract affect the animal itself, whether it's epigenetics type of activity or others, we don't know yet.
Thank you.
Yeah, from INRAE. Perhaps a little bit general question. Why hasn't nature invented a proper mitigation strategy? Not so much for global warming, but for the ruminant, because ruminants eat poor quality feeds compared to monogastrics. They teamed up with microbiota, including methanogens, that induces a loss of energy. Why haven't ruminants evolved with a more, I'd say, energetically efficient microbial ecosystem?
It's extraordinary question that of course I don't have the answer. But you have to think that, well, the ruminants, they take most of what they can from their microbiota, and is garbage or is a tax that the methane, I don't know. But in any case, what is important also to say is when you reduce methane emissions by using additives, that energy that doesn't go into methane doesn't go either into the animal production. So there is well, that's important also for policymakers, and there's a lot of try to sell things saying, well, we will reduce emissions and we will improve production. We don't see that. So we have to be careful also with that message.
Thanks, Diego. Sinead Leahy from New Zealand. Did you follow, two questions. One, have you followed the animals? Or do they still exist? And have you thought about going back to measure and see how they look now? And two, also, so you showed that there was differences, you could clearly see differences in the microbial composition, but, do you understand why you had the lower, why they were emitting lower? Was there less abundance of particular methanogens, or were the methanogens functioning slightly different in terms of their expression, And yeah, just your thoughts on why you think it was low emitting. Yeah.
For the first question, we managed to get them when they were close to 1 year old, and after that we lost them because they were used in other experiments, and so the trajectories of those animals, I don't know. So there were not very many as well, so to draw conclusion is not that strong. For the second question, we don't know. There were no differences in the biomass, if you measure by qPCR on bacteria and archaea. The only thing that we know that there were different, but up to find the particular microbe that we can say is responsible for those changes, no, we couldn't. But as I said before, there's changes in the microbiota that persist, but are they the reason why we observed that small changes in the methane emissions? I don't know either.
Thank you. We'll take two last questions, but please be brief so we can soon go to lunch.
Mariano Parra from the University of Queensland, Australia. So my question is, you didn't observe changes on BFAs, but you observed changes on methane. Where do you think the hydrogen went? Or do you think there is a risk of hydrogen accumulation, especially that there are some researchers that mention that high hydrogen accumulation can affect rumen fermentation? And I have another question. In a long term, what is the impact of inhibiting methanogens community? Would be great propionate production, and how about the acetic production, as high accumulation of hydrogen can reduce the acetic production. I mentioned this because in extensive grazing production, acetic is quite important for animals. Thank you.
Yeah. Again, I don't have the right answers to that. It's true that we didn't see any differences in BFA production, which is kind of surprising, but we don't know why. We don't know why.
Yeah, thank you. Before lunch, I give this simple question. I think early life intervention is. I'm Sang-Kwon Ro from Japan. I think early life intervention is very useful and effective technique to reduce the methane emission. So, but I think the point is very important. I think the first. The 3 NOP is just used for 6 weeks, but I think if more reduce the time and more increase time is more the effective to reduce the methane emissions. So, especially the before weaning, you treated some NOP is more effective. So, what do you think about which point is more important in the reduced methane emission?
I'm not sure. Can you reformulate the question? I'm not sure I understood. Yeah. You are talking about the length of time that we applied.
I think the time point is very important. Or afterwards, the first 2 or 3 weeks is more important, I think.
Yeah, okay. I don't know whether I got it, but we couldn't test in different application times. Personally, I think the first 3 weeks are very important. And other studies have shown that there's a change in the microbiota at about 3 weeks, which is the time when the animal start eating more solid foods. So that window of time is very important. After, I cannot say whether if you do just 3 weeks, it will work or not.
Thank you.
Thank you, Diego. And thank you again to all the speakers. And this closes our first session for this morning. Philippe?
Yes, just to say that we will start again at 2:30 this afternoon for the biodiversity session. So we are going to have the lunch in the same room as a coffee break this morning. I hope that it will be quicker. So we start again in the same room here at 2:30. Okay?